Welcome to our pH meter experiment for General Chemistry 2. The first step to using a pH meter is to calibrate the equipment to correctly measure the pH of your solutions. You'll use a solution with a pH of 7 and a pH of 4, rinsing after each measurement of a different solution. Then enter your corresponding pH value to the tested solution into the provided box and press Keep. You'll repeat this process for both solutions. In order to verify that your pH meter is correctly calibrated, test your known solution of pH 7 and make sure it is reading a value of 7. Obtain the 6 solutions and record the recorded pH values for all 6 solutions in the table provided. Be sure to rinse with distilled water after each test. You will use the M1V1 equals M2V2 equation to correctly dilute the 4 different concentrations of HCl and the 4 different concentrations of acetic acid needed for the exercise number 2. Using the results from the M1V1 equals M2V2 equation, you will then use separate pipettes to dilute each solution using your stock solution of acetic acid and HCl. Remember to use a graduated cylinder to correctly measure the needed amounts of water and needed amounts of stock solution to correctly give you the necessary dilutions needed for experiment 2. The main difference between a strong and weak acid is its ability to fully or partially dissociate in water. A strong acid will fully dissociate, while a weak acid will only partially dissociate in water. Again, record and compare the pH values of the diluted solutions to their corresponding concentrations. This recorded data from this experiment will allow you to create a scatter plot diagram that illustrates a relationship between concentration and pH of that solution. In order to create the buffer that is needed for exercise number 3, you will need to mix 30 milliliters of a 0.02 molar solution of sodium hydroxide with 60 milliliters of a 0.02 solution of acetic acid. Also, create a separate beaker of 30 milliliters of water in order to compare the effectiveness of your created buffer. A buffer solution acts as a proton acceptor, or H+, which can in return resist a drastic change in the overall solution when an acid is added. This will be displayed in the data received in the next slide. Finally, record the pH values of the created buffer solution and the water solution when HCl is added to the mixture and compare the recorded data. In order to correctly clean your lab bench, you will need to carefully return your pH meter to your TA and dispose of all paper material in a trash can located at the end of each lab bench. Then, clean and return all used glassware to their proper locations. This will conclude the pH meter lab experiment.